First presentation is by Dr. Stephanie De Vos on Artemia as a model organism for epigenetic research in aquaculture. Stephanie, you have the floor.
I'm Stephanie De Vos. I work for the Department of Applied Animal Science and Welfare at SLU Sweden. And I have worked on Artemia most of my research life. So what I will talk about today is why we need models in aquaculture research, what is a model organism and what would be a good model for aquaculture. Also, what's special about Artemia biology, why would it be useful as a model, and then also why Artemia would be a good model. Then a little part about genetics and epigenetics and how we can apply these technologies in the Artemia model. And then finally, the benefits of using Artemia as a model for IAAC and what the future would look like for Artemia as a model. So first of all, why do we need models? Crustacean research is incredibly expensive and the cultured species have a very long life cycle. And moreover, aquatic species are an abundant animal protein source and they require less feed crops and land than terrestrial species. Nevertheless, most of the sequenced genomes are in terrestrial species and the sequenced genomes in aquatic species are only recently growing, relatively recently. So why do we need models with sequenced genomes? A sequenced genome allows you to determine a lot of things. You can determine the large-scale genomic variation between individuals. You can determine traits for other species that are similar. You can identify markers and genes responsible for a trait, influence those traits with genome editing and RNAi, do breeding or selection, control pests and invasive species, and help conserve species, which would definitely be interesting for Artemia. And finally, of course, this type of research is interesting for fundamental science as well. Crustacean models are needed because right now the most used crustacean models are Daphnia and Lithobenese vaname, the two animals you can see here. And the number of crustacean genomes that is available in current databases is 134. Now I'll have you know that the amount of crustacean species is estimated to 75,000. So that means they are actually not sure. Huh? They haven't searched the whole ocean, but the estimation is 75,000. So that means that 0.18% of crustacean genomes is sequenced and that crustaceans are an incredibly under-researched group. But the good news is that Artemia is one of the few sequenced genomes of crustaceans. And this is relevant for IAAC because it gives us more knowledge about Artemia and how we can make sure that Artemia biodiversity is conserved. So Artemia is a lesser known model, but highly useful for aquaculture, and I hope to convince you why. So just a definition, what is a model organism? A model organism is a species that has been extensively studied to understand a particular biological phenomenon, which could be a trait or a disease, and with the expectation that discoveries in the model will give you insight in the working of another organism. And why can we derive insights on other organisms from one model organism? Because evolution. So there is conservation of biological pathways and genes because animals have a common descendant. And another few traits that model organisms should have is that they should be suitable for lab studies, they should have a short generation time, a characterized genome, and they should be similar to important species. Now, why would we use a model? Because sometimes there are ethical reasons. A model organism is often used as a replacement for a higher animal. just in the way that Drosophila is used for human research, and is also often used as a substitute for more complex or less studied animals. And there, Artemia could be useful because a lot of crustaceans have been much less studied than Artemia. And also, using a model can be cost-effective because it shortens the duration of your research, because the generation length of the model species is shorter. It's easier to do controlled studies because a model organism is easy to breed and small, and the genome is usually smaller, and that means less data to process to do any kind of analysis. And finally, we also expect a model to have available genetic tools such as a genome, a transcriptome, maybe some RNAi, mutants, genome editing. So what would be a good model for aquaculture? There are a few criteria called the Davidson criteria to select a good model. It should have the part of the biology that you want to research. We're talking organs, tissues, and also it should have a similar pathway or biology in the biological process that you want to study. And also a good model should be able to be validated in the organism of interest. For example, if we use Artemia as a model, as a model for crustaceans, we should be able to validate that the process we're studying is similar to that in crustaceans or in humans. And a final take for models, no single model fits all questions, and so you should select your model based on your specific scientific question. A known aquatic epigenetic model is zebrafish. So zebrafish has been shown to have an epigenetic role for its response to environment and more specifically disease development, like development of cancer, obesity, diabetes, aging. And these mechanisms are mostly regulated by methylation mechanisms in zebrafish. There is also some epigenetic inheritance. And moreover, zebrafish, mostly also in genetics, but also epigenetics, is known for studies on embryonic development because the embryo of zebrafish is transparent. So development can be studied closely. Some Artemia biology. What is interesting about Artemia? Why do we want to use it as a model for crustaceans, and or for which biological processes could it be a model? So first of all, Artemia has unique biological features. It's an extremophile. It thrives in extreme hypersaline environments. Its life cycle is short, and it has several reproductive strategies depending on the species, like bisexuality and parthenogenesis. And it has diverse habitats because it's adapted to extreme environments such as salt lakes, but even those are quite different, which gives rise to genetic diversity and so becomes an opportunity for genetic selection. Artemia also has an ecological importance because it has a role in aquatic ecosystems, and there are threats to its biodiversity and its habitat degradation, as you all know. What makes Artemia a good model? Currently, it's already used as a model for toxicity studies, disease resistance, because Artemia can be grown gnotobiotically, so that means that you know which microorganisms are present on the animal, and in this case, of course, it's like a sterile culture of Artemia. Also, it's been used as a model for crustaceans and as a model for parthenogenesis. It's easy and cheap to culture. The brood is quite large. It's easy to replicate, and it has a short life cycle, of course, about 2 to 4 weeks. Also, there are genetic resources available. The ARC cyst bank is known to many of you, where you can find different species and strains. Also, inbred strains are available, clonal populations. There are several genetic and epigenetic tools available for Artemia, such as a characterized genome, transcriptome, proteome, a detailed genetic map. The RNAi technique has been mastered for several genes in Artemia. It's been transformed, so mutants have been made for Artemia by electroporation, and there have been transgenerational epigenetic studies. Moreover, there are embryonic cell lines available, and these cell lines and the availability of different types of cell lines is expanding. And then finally, of course, Artemia has a unique experimental versatility. That means you can do common garden setups with cysts that are from different generations. That's quite unique. And also, it's possible to have axenic culture, so sterile cultures. Here you can see all the different publications, and it's just a selection of all the different genetic and epigenetic tools that have already been developed for Artemia. So we're talking about RNAi, genomes, transcriptomes, genetic mapping, and transgenerational epigenetic studies. We hope to make, just like Arabidopsis is a plant model for maize, we hope to make Artemia a crustacean model for animals such as Vaname, because Artemia has a much shorter generation time. You can see that the generation time of Artemia is about a month, and that of Lithobenese vaname is about a year. Also, it has a cyst bank, which doesn't exist for Lithobenese vaname. It can be grown cheaply in bulk at a fraction only of $1 per Artemia, where Lithobenese vaname at the end of the road costs almost $2 per animal to grow, because the feed needs to be adapted, there needs to be disease management and water quality management, and those things are much more expensive for Lithobenese vaname. And also the market of larger shrimps is billions a year. So it can really be interesting to develop Artemia as a model for that. So what is a genome? It's all the DNA in a biological sample. The sample could be an organism, tissue, cell, organelle, and this includes all genes. So it includes actually all biological information of an organism as a blueprint. Genetics researches the function and composition of one gene, while genomics, and that's actually the type of research that interests me, genomics researches all genes and their interrelationships to identify their combined influence on the organism. There you have a much better image of what's happening on the whole. Currently, in genomics and genetics, there are a few developments, such as the development of MinION long reads. This is a type of sequencing machine that can be brought to the field, that is as small as a cell phone, and that you can connect to your computer. And a normal laptop is actually enough to do the sequencing. And at SLU, we have been doing MinION sequencing for genotyping of different Artemia samples. So this is actually the technology. The DNA strand goes through a protein that gives an electrical signal that you can see on the right. And that electrical signal differs depending on each base in the DNA sequence. And then we can actually have a data file with the sequences. So what is an epigenome? It's all the chemical or spatial modifications of DNA that regulate gene expression in a biological sample at a given time. So as you can see here, you have a chromosome. The DNA is wound up around proteins called histones, and the very tightly wound DNA strands have a modification in gene expression, Same for DNA, where methyl groups are attached. That also influences gene expression. So that's mostly what happens when we are talking about epigenetic processes, is that the environment influences the winding of the DNA around the protein or influences the methylation of the DNA. And that influences the expression of a certain gene. And so when we study the epigenome, we get a snapshot of gene expression regulation in a sample. So how can we apply genetics or genomics and epigenetics in the Artemia model? First of all, we can do some extreme stress response research about toxicity, pollutants, heavy metals, anoxia, high salinity, high temperatures, pH, radiation, and pathogen resistance, such as for Vibrio. And it can be applied for aquaculture to model genetic and epigenetic responses to environmental stressors in aquaculture species. Also interesting is research about diapause and quiescence. And there are quite some applications for human health. So Artemia is already been used as a model for cancer research. And it would be interesting to test genetic and epigenetic regulation of quiescence and dormancy pathways. Also, could it be possible that unique Artemia compounds can induce human diapause, such as for space travel? There have been some publications exploring this concept. Benefits for IAAC in Artemia as a model is, first of all, there can be aquaculture applications, as I said, to model responses to environmental stressors in aquacultured species. More knowledge about Artemia stress response in different strains, such as the response to climate change and environment change, invasive strains, and the potential for strain selection of Artemia. And finally, a model organism gets much more attention from society, which would give rise to better opportunities for conservation of Artemia species and strains. And I mean, in the sense of for aquaculture or as a sustainable human food source, but also more funding for Artemia research. So what would be the future of Artemia genetics and epigenetics? First of all, we have all noticed that AI is taking growing space in our lives and into the media. So AI will be involved in it. Single-cell sequencing, which means sequencing of different tissues and cells separately. Genome editing, increasingly long reads. Also using multi-omics, not only looking at DNA and RNA, but also the proteins, the metabolomes. And then also more and more genomes will be sequenced of different strains that give us a complete view of what Artemia actually looks like. And finally, there is even a company called Colossal. They sequence genomes of animals that are extinct, but also of existing animals, with the idea of when they get extinct, we will have the genetic information and we will be able to de-extinct them. We will be able to revive them. I'm sure we've all seen Jurassic Park. So maybe it would be interesting, just as a concept, to characterize genomes of Artemia and that way have all the genetic information so that we can revive them if necessary. I see that the time is almost there. So the future of Artemia as a model is, it has global relevance. The role of Artemia is important in addressing emerging challenges in aquaculture, climate change, cancer, and food security. My take-home message is Artemia has unique advantages as a model organism. There are potential applications in epigenetic research. It's relevant to aquaculture, sustainability, and conservation. And model organisms receive more media coverage and funding. Just think of tardigrades. Tardigrades have been in the news because they can withstand radiation so much, but Artemia is more resistant than tardigrades in many ways. So it could be useful to put Artemia on the market that way. Thank you.
Thank you, Stephanie. Meantime, you can. I think you have convinced us that Artemia is a good model organism, and I like very much also the way at the end you have made a plea, of course, for further research funding, but also to mention that we have to give more social attention to Artemia. And last week at the FAO meeting, this was also one of the conclusions. We do not realize, okay, we operate within our circles of the convinced, but what you have shown here, we should bring it to a wider audience. Thank you very much.